Enzymes. Speaking of which, I just ate a Reese's peanut butter cup. So I should have thought before I hit record. Sorry, guys. But guess what's breaking down my starch? Saliva, which is an enzyme, sweet transition. So enzymes, one of their main jobs is relating to energy. And I'm not going to spend a lot of time on having you know exactly what I mean when I talk about energy, but what I do want you to know is that energy means doing work, whether it's synthesizing molecules, making molecules, moving things, generating heat. Our body has to do all that. And enzymes facilitate that. So you've got your kinetic energy, the energy that's released, and potential, which is the energy that's stored. So when we talk about energy, we also have to talk about metabolism. In relation to enzymes, we talk about them forming bonds and then breaking bonds between molecules. So when they form bonds, we call that the dehydration synthesis because water is being released, right? You're making something. So D is getting rid of, hydration, we're talking about water, synthesis, putting together. Enzymes also help us break bonds, in particular the hydrolysis reactions. So like digestion, right? We break bonds. Enzymes break things down for us. For example, here's your dehydration reactions, your synthesis. So what we are missing, I'm going to try to draw this in for you, is, there we go. Water is released, water is used. That makes sense. So the dehydration, water is released when that bond is formed, and the digestion, water is actually used to help bring that up, if that makes sense. There we go. So you got your enzyme being released. That could be the formation of like your sucrose, right? So your sucrose is right there. And so you have water actually being released when that sucrose breaks up into glucose and fructose. And again, you've got water being used to help, I'm sorry, I said that wrong. When those sucrose and fructose up here, when they're combined, water is released. Down there, digestion, when they're broken apart, you need to add that water because you're forming two new molecules. That made sense. So where do they come from? Where does this energy come from? Well, it comes from the work of the enzymes, either breaking apart, which is releasing energy, or putting together, which is using energy. So I like to think of enzymes as like party accelerators, right? You're having a hangout, maybe you're fructose hanging out with glucose, right? Not a whole lot going on. Enzyme shows up, kicks the party up to a 10. So you're talking about enzymes are proteins. And what they do is they lower the activation energy. They lower the amount of energy needed for a certain reaction. So they help to speed things along. And they're the ones that are basically in charge of our functions, right? Whether it's digestion, our heart beating, us being able to regulate our temperature, all of that growing, all of that is because of enzymes. So the three main parts of the enzyme process is this: the substrate, which is what is the reactant to which binds to the enzyme. This right here is your substrate. Your product, which is going to be the end result. And then your active site, which is the site that bonds, okay? So substrate is what it is kind of like beforehand. The active site is where the substrate binds to the enzyme, and whatever is released are your products. A couple properties I want you to be aware of when it comes to enzymes. First of all, they're specific. The enzymes are very specific to a certain type of substrate. So if you have like a lipase, right? A enzyme that only breaks down lipids, it ain't gonna party with carbohydrates or proteins. It only wants to party with lipids, lipase. The other thing about these enzymes, they're not consumed. Once they facilitate a reaction, they can just keep on keeping on. They can just keep on facilitating more and more reactions. And the other thing is they are affected by their surroundings. So depending on a number of factors, that could definitely impact how a enzyme is able to function. Okay, couple things. So under naming, I want you to know that enzymes are specific to, like we said, they're specific to the certain substrate or reaction. And what that means is they, generally speaking, end in, they end in ASE. Sucrase breaks down sucrose. Sucrose is a sugar. Protease breaks down proteins. Lipase brings down lipids. Polymerase breaks down DNA. Okay, now pepsin is kind of this weird one in your stomach that breaks down peptide bonds. But pep and pep. And there's just kind of the reaction like we saw before. The substrate sucrose bonds to the enzyme. The enzyme is sucrase. Okay. Water, because this is you're getting two products. Water is used. Okay. And then your products are released, glucose and fructose. And then that enzyme lives to facilitate again. Okay, it's not like a mosquito when they sting you or a bee. When they sting you, a lot of times they die. Not so much with enzymes. So think of it like a lock and a key. You know, you've got your key that fits a specific, or you've got your lock that can only work with a specific key. That's a lot like your lock would be like your substrate. Your lock would be your enzyme, and your key would be your substrate. And they work together to create a reaction, or in this case, they open your door or your whatever. So again, it's the key here is they want to lower the amount of energy used in a reaction. So how does it work? It works because either they combine things or they digest things. So enzymes can either bring things closer together or they can separate. That's how they work. That's their main function, okay? A couple things that affect the enzyme function is going to be how much enzyme do you have, how much substrate do you have, temperature, pH, or inhibitors, if there's anything in there trying to get in the way. For example, temperature, right? Say for human enzymes, for us, you want to be our body temperature. So this is 37 degrees Celsius, 98 degrees Fahrenheit. If it goes above that, so if you have a fever, it's going to impact the enzyme's ability to do its job. Again, if it gets lower, same thing. Your molecules are going to move slower, and the amount that enzyme can do is going to slow down. Here's the thing, though. If you, with the heat increase, you also run into what's called denaturation, which basically means it changes the shape. So let me see if I want to write that down. Oh, there we go. It changes enzyme shape. And we all know that enzymes are proteins, and proteins' ability to do their work depends on their shape. Okay. And again, if you decrease the temperature, they're going to just move a lot slower. pH, right? So again, if you change the pH, if you make it a more acidic or more basic environment, it's going to change the structure, right? It's going to change the structure, which changes the protein's ability. Again, we're looking for most enzymes, we're looking for neutral-ish. Now, is that true for everyone? No, because your stomach is a very acidic place. So we know that pepsin, for example, and we'll talk about pepsin later when we talk about digestion, is you need to have a lower pH. It has to be acidic. Trypsin, which is going to be in our small intestine, needs a little bit higher pH, a little more basic environment. Okay, guys, I know I went really fast. I kind of wanted to cover the basics with you. So what I'd like you guys to do is bring in your questions tomorrow. If anything was unclear or I went too fast, great. I'd like to, I'm going to give you guys like a little tiny quiz on this one. It's like a 5-point quiz. But before I give you that quiz, I am going to give you guys a minute to answer or to ask any questions you have. So please bring those in. So I can just make sure that I am making sense, and that's all I got. Also, if the Cubs do not win tonight, I'm going to be very, very grumpy. But I'm going to make your quiz before that, so it will not affect your quiz. Okay, guys, thanks.